But I promise right after this session, we're gonna have a nice lunch break. So I just wanted to say we are going to get started with the next session that is focused on neonatal seizures, which anybody who is into neonatal drug development would understand it's a really complex and challenging issue. The original moderator for this was supposed to be Dr. Mark Turner. Unfortunately, who couldn't be here today. So I'm basically just taking over for him. I would like to invite our next set of distinguished panelists, Dr. Ann Massaro from FDA, Janet Soul from Boston Children's Hospital, Carol Kenner from Council of International Nurses, Betsy Billon from Hope for HIE, and Ashley O'Neill from It's a Nicky Word. Thank you, the panelists, and we will just get started in a few minutes. So as an introductory remarks, seizures are one of the most common and most challenging complications in neonatal brain injury. They have, you'll find them in multiple different contexts, in HIE, in whole other neonatal brain injury type of situations with long-term sequelae. Despite their frequency and impact, progress in neonatal drug development has been extraordinarily slow. I would say, slower than much of the other neonatal drug development even. It's constrained by challenges in defining meaningful endpoints, standardizing EEG monitoring, navigating, ethical and operational complexities of trial design, and the fact that there's already a kind of a gold standard treatment available, that is phenobarbital, that makes downstream drug development even more complicated and challenging. So we have a work group within INC. We call it as a seizures work group. Uh, but then it basically has built on foundational efforts to move the field forward when it comes to addressing the challenges in neonatal drug development for neonatal seizures. We want to start thinking about and establishing next level strategies, including bridging clinical EEG imaging data. Right now we were just having discussions and publishing manuscript, but this is not where we really want to end up with. We want to lay the groundwork for regulatory grade enrichment strategies and endpoints that would make neonatal seizures drug development more feasible, interpretable, and ultimately, at the end of the day, more impactful for the families. So we are going to have a really good, hopefully, nice discussion for this panel session. And ultimately, I would obviously encourage audience to ask any questions that if you might have, given how under-addressed the science of neonatal seizure drug development is. So I'm actually going to get started with questions. So Janet, I'm just going to start with you. From, I guess, with an academic hat on, you work at Boston Children's Hospital. From your perspective, what is the most promising way to define seizure burden as an enrichment tool or as an endpoint? What, in your perspective, is the most clinically meaningful aspect of that? Frequency, duration, cumulative burden, and how does this differ from what is being done clinically in the clinic versus somebody in industry might want to approach this as a potential drug development?
Okay, so that's not the $1 million, but now multi-million-dollar
Yeah,
question. That we have
absolutely.
quite spent at least that amount of money
Yeah.
trying to study that. So, there are not great data showing, what level of seizure burden, whether it's frequency or severity of some kind, whether it's episodes of status epilepticus or whatever, is most important. And so that has been a very vigorous argument in our community about how should we design trials to, in terms of defining an outcome. And, sure, you would like to say seizure cessation would be ideal, but maybe that's not necessary. And I think that gets away from the main question, which is what's the goal of reducing seizures? Really, what we want to do is have less epilepsy and less neurodevelopmental disability or impairment. So we think that if we treat the seizures more effectively, we may accomplish those two things. We have not yet proven that. We haven't we certainly have a lot of evidence from the animal data that more seizures beget epilepsy. We have some data from humans that children with more seizures as newborns are much more likely to have epilepsy, much more likely to have later impairments. What we don't know is how much we need to reduce neonatal seizures to improve that long-term outcome. So that's the real key question. It's not about how many seizures should a baby have. It's like, what do we need to do to make them live a better life without epilepsy? So my dream is that we prevent epilepsy in the babies with acute symptomatic seizures. And so I say that specifically because there are two different kinds of seizures, broadly speaking, in newborns, for those of you who don't work in this area every day like I do. There are those seizures produced acutely by some type of perinatal brain injury, whether it's HIE, the most common cause, or other causes such as stroke or hemorrhage or infection. Then there are those babies who have epilepsy that just happens to start as a newborn. So they have a brain malformation or genetic disorder. Those kids are very different, right? Those two groups are very different. So it may be that if we have a better treatment for neonatal seizures that are caused by brain malformations, that those kids also do better in some way, but it is very different to measure those two groups together. So although we might have early phase trials where we test drugs in all comers with seizures, when we're talking about our long-term outcomes, and for example, my dream of preventing epilepsy in babies with HIE, um, you know, we need to study those groups separately, or at least analyze them separately in our clinical trials. And so I think that's the really big challenge. Uh, there have been a ton of papers showing that more seizures lead to worse outcomes, and we don't have a great sense of where that threshold should be. There's exactly one paper that said 12 minutes per hour, and yet other papers have not found a threshold. So I don't think we should just hang our hats on that one paper with 70 kids or some number that really doesn't amount to a statistically significant finding. So maybe a bit of a long-winded
No, I think
answer, but at least I tried to set the scene for what the problem is.
it's a really important issue. I mean, especially that all of the standards have been based on just one paper, and with 70 patients, it's kind of shocking. But that's the way things are. Um, you know, um, I'm gonna direct my attention to our patient members, parent and members on this panel. To Ashley and Betsy. From a family perspective, who have had either like children with HIE, or kids who've had, like in your case, Ashley, neonatal seizures. What is, in your perspective, what is it, the one thing that matters the most when it comes to neonatal seizures and neonatal seizure drug development? Is it seizure reduction? Is it like ultimate outcomes, developmental progress? Is it quality of life? Is it all of the above? Is it none of the above? What is it that matters the most to you? So maybe Ashley, I'll ask you to answer the question first, then I'll go with Betsy.
So for me, it is all of the above. It's the quality of life overall. It's the peace of mind. Currently, my son has seizures, and whenever I am going to wake him up, I quickly pause, say a prayer, and like, am I going to walk into him having a seizure?
Mhm.
So it's all about, and then he's, 6 in first grade. So you're wondering like his overall developmental ability. So it's all of the above, quality of life, peace of mind, the ability to just live a normal life and
Mhm.
not, we're heading to church the other day. We have to turn around because he's having a seizure in the car.
Okay. So do you want to talk a little bit, it's unprompted, not in the
That's
list of the questions, but tell a little bit about the story of your
my son.
child, Colin, and,
Mhm.
so that people understand the context where you're coming from.
Sure. So my son was born at a small community hospital that didn't have the resources to properly intubate him. So he received manual ventilation for an hour until the flight team from Georgetown came and flew him to DC. He has developed bilateral brain bleeds, grades 3 and 4, 2 cystic PVLs, and shunted hydrocephalus. And so we were told all of the scary things that will possibly happen. Some of it did happen. But he's 6 now. He's non-speaking, but bosses me around with the AAC device. He could spell better than most first graders. So it's just been a journey of finding that balance of, giving this kid the best possible life, but also managing his multiple comorbidities.
Right. And in your perspective, the treatment he received or the lack of treatments, has that impacted you, Colin? How that has impacted? I mean, I really want to focus on the human side of the story here.
So when he was in the NICU, he only had one seizure. And so they hooked him up to the EEG, and they was like, well, everything, the electrical activity they're showing is not enough to classify that he's having seizures. So shortly after he was discharged from the NICU at 6 months of age, he was taken off all of seizure medication. And then one day when he's a year old, I go to work and my husband sends me this video. He's having a seizure. And then fast forward, he went 2 years without having seizures. And then from like 2 to 5, it was quarterly. And then it ramped up to weekly. And so now we're finally at the mix where he's on 2 medications, so what, triliptal and valproic acid. And as we are increasing the doses because he's having breakthrough seizures, now you're worried about the other side effects, like, oh, now his platelets are dropping. So that's adding like more blood work. And then now the neurologist just was like, oh, well, maybe we need to go get a VNS. So it's kind of like, should more have been done when he was in the NICU, or to kind of predict that he's going to later start having these seizures? Because he was seizure free for like 2 years, and then all of a sudden it's just like, bam. And you're like, how can you live like this?
Yeah, no, that's definitely terrible.
Some of the seizures were lasting upwards of an hour. And so then you're getting flown up to Georgetown, and we live 2 hours south.
Betsy, do you want to talk about it in your perspective? You've worked with HIE community for so long, even though I know you told me that your son did not particularly personally get impacted. Maybe I'm just wrong with that, but you live this thing day in and day out, right? So, from your perspective, what is it that matters the most to you?
Sure. So just for a little bit of background in both the advocacy work that I do with Hope for HIE and my personal story. So my son Max was born just over 13 years ago with HIE delivered into our local community hospital that I had actually worked for the health system in marketing and communications. He stopped moving at 37 weeks. So we went in expecting to get checked out and leave like everyone else we had heard of with that experience, but that was not our experience. And so he was delivered by emergency C-section. And he was bolused with phenobarb before we even got transferred to the higher level care down at Henry Ford Hospital in Detroit for cooling. And so we were very fortunate to have exceptional care of the same neonatologists in the health system travel through the community hospitals for continuity of care. But Max didn't have any noted neonatal seizures, highly likely because he was already bolused on phenobarb and continued that. His EEG during cooling was very flat. They were concerned that, things were really, really bad. But as he started to wake up and, maintain the phenobarb dosing, he was, able to have a better clinical outcome than I think everyone had initially hoped, which is great. But also the long-term story of this, of my own personal story, as some of you may know, is Max developed epilepsy at age 8. So well out of the range of what we had been counseled. We have a wonderful neurologist who happens to be an epileptologist as well. So and he is self-proclaimed as being less aggressive in, follow-up EEGs and things like that. Because, he always said, like, he's probably going to have an abnormal EEG his entire life, regardless if he has seizures. And sometimes the insurance companies will want you to do something about that. And we don't want him to be on, anti-seizure medications unnecessarily. So, okay, understandable. And we just don't have a lot of good long-term data for seizure burden in HIE and things like that. But, when we come to the community aspect, and what's really powerful is, we have now connected over 10,000 families globally since the time that I joined the initial Hope for HIE Facebook group with 200 families. And we know when we talk about small cohorts, right? When you look at these larger cohorts, this is not an unusual situation of families, of kids. We know that there's this group that's going to have persistent seizures out of the NICU. We know that there's going to be kids that develop epilepsy in the first couple of years. And this is like kind of this gap where families are coming to us weekly in our community of age 5, age 8, 10, 12, 14. And saying, wow, we thought we were out of the woods for this. There's no predictive modeling currently that's doing, we're getting there with the neonatal seizure registry and all sorts of great work with that's going on. But epilepsy is a huge burden, as Ashley mentioned, Max's seizures are nocturnal. And so many of our kids have these aggressive nocturnal seizures. And while, knock on wood, he hasn't had one in 2 years now, which is incredible. We're so thankful for that. It could happen at any time. And, that fear is real. The quality of life for the children and families is real. Max doesn't get to go to sleepovers. He doesn't, I have a video monitor that my husband is the only one. He's freaking out the next 3 nights. Thank you for, taking on all the parenting duties while I get to be here. But we have a video monitor that's a seizure monitor at night, every night. I'm always terrified that I'm going to wake up and my child is going to die and be a victim of SUDEP, regardless of him being pretty well controlled and all these monitoring devices and things. That's just the reality that our families live with. So we talk about the tricky thing with neonatal seizures and anything with epilepsy, really with neonatology, is you have this acute time period and then you have everything afterwards. Excuse me. And how do we develop and have these biomarkers that help us take that unknown and that long term, which is more than age 2, age 3 that these trials go to, that have these impacts for this community and any other sub community, as Janet articulated as well, and really figure out what we can do to decrease that.
Well said. Thank you, Betsy. I guess coming to Anne, FDA, Anne, you've heard that, it's not just original question was considering seizure burden, but then, you heard from um, uh, uh, from Ashley and from Betsy as well that, it's not just one aspect that matters, not just original like seizure burden in the neonatal period. That's the only thing that matters, but all of the above, seizure burden, seizure reduction in the neonatal period, prevention of long-term occurrence of epilepsy as a consequence of whatever, long-term sequelae of whatever happened in the neonatal period, developmental progress, quality of life, all of those things. What sort of evidence would FDA and regulators would really need to consider those as credible and or important endpoints to be used in clinical trials?
Thanks for the question. So I think first off, I think a lot of that, was really hashed out with INC work group 1.0, to go through and get all of that multi-stakeholder, from all disciplines to kind of talk about, if we're going to look at an anti-seizure drug, then probably the efficacy endpoint that makes sense is reduction of seizure burden. And there was a lot of work that went into making those recommendations. Thanks to Janet for leading that charge to come up with some consensus guidelines around, what makes sense to use from a seizure burden reduction perspective as a primary efficacy endpoint for these types of drugs. And I think we've seen with the approval of phenobarbital, there's somewhat of a path now forward with an approved drug and trials that have been well done to, from the primary efficacy endpoint perspective, I think have somewhat of a regulatory path for that. I think that what you're talking about and what we're discussing here, of course, that's not the only important endpoint here. And whether, depending on, when we talk about seizure drugs or when we talk about HIE in the next session, all of these other outcomes that are really important. Of course, the multi-million-dollar question is, what degree of seizure burden, relates to reduction in epilepsy, relates to improved neurodevelopmental outcomes. And then even beyond that, if you can reduce seizure burden in the hospital, get a baby extubated earlier, reduce length of stay, then, again, hearing from families that that's an important endpoint as well. So all of these things, I think, are important. And I think, depending on how the drug works, there's not a single, path for a whole class of drugs. I think it depends on the specific drug we're talking about. In this context, anti-seizure drugs, we want to see that they stop seizures and that these other endpoints, of course, are important when we talk about neuroprotective agents in the next section, those kinds of things. And I think a lot of these endpoints that we're talking about are very clinically relevant. And I think when sponsors come in and propose different endpoints, propose, primary and key secondary endpoints, it's really important for us to to consider those and hear. And I'm really glad that Betsy and her group has really been involved with a lot of different drug development programs and a lot of different sponsors, because for us, hearing that patient perspective and that sponsors have consulted with patients and have that incorporated that into what they're proposing for their primary key secondary endpoints are something that we very much, consider and think about from the regulatory perspective.
Great. Thank you so much, Anne. Coming to Carol, nurses, to my understanding, because a lot of clinical trial enrollment that needs to happen in neonatal seizure trials need to happen within hours of the birth, right? In NICU, and more than I guess physicians, neonatologists, nurses are basically the primary first point of contact with the families. Um, Patient enrollment, patient engagement, and making sure that they agree to participate in those clinical trials is a crucially important consideration. But in your experience, what do you think are the most important or significant or practical barriers that that you've seen within the NICU settings that nurses and patients face in this neonatal seizure drug development or clinical trials?
I'm going to say that it hasn't changed, unfortunately, a lot in the last few years, even though through this group, I think there's been a lot more awareness of including and engaging nurses in the very beginning in discussion of protocols in the clinical area. But that's not true. I work with nurses globally, and that's certainly not true globally. And why do I say that? The barrier these days is because of the severe nursing shortage and physician shortage in many countries, and so time. The nurse that's providing the care and the scheduling of the tasks that are important in the care that's vital is also competing with sometimes the scheduling of the protocols for clinical trials. And I've advocated for a long time that if it was possible for nurses to be involved, even in the very beginning of the development of those protocols, you'd find that there would be less competing of time and less friction when it comes to why was the data not collected or why was the blood draw not in a good schedule with the normal rhythm of the unit. That's number one. I think the other thing is both parents in particular said, and certainly working, and this came out in the nurses paper with Mary Short and with Deb and others, the nurses are the first line where parents ask the questions. I've heard so many times over my career, I don't want to bother the doctor. I don't want to disturb them. But can you explain why I should enroll my child in this study? Not that the person that's trying to get informed consent doesn't do a good job, but at least in my experience, having served on a children's hospital IRB for 12 years and seeing the protocols and all the things that you have to read before they can sign for informed consent, we use the jargon, but what does it really mean? And nurses are at the forefront of trying to explain that. But if we weren't included in understanding the protocol, if we don't understand exactly what this trial is all about, then how do we explain it to the parents? And that's what makes me sad, is that I've been doing this a long time, and I've been with Inc. since the very beginning. And we're making progress, but we're not there yet. And nurses really want to help the parents understand and make informed decisions. But then if we don't have the knowledge and the understanding of what is going on, then that's an impossible task. And I would also say that we haven't done a good job in a lot of our nursing programs to talk about long-term effects. In other words, after you're out of the NICU, what should we be working with as we work with families in the community settings and follow-up clinics? How can we be supportive in that role? So I think that's another thing. So information, inclusion, and certainly time. Those are the barriers I think that we have right now.
And from, Betsy and Ashley, from your perspective, I believe, Carol talked about her perspective coming from a different angle, but maybe things that you've heard from your community members and maybe, Ashley, in your case, when your son was in NICU, did you have any issues in terms of, knowledge of a research program, importance of participating in a clinical trial or a clinical research program, and your baby was in the NICU. Was there any issues pertaining to consenting and agreeing to participate in those clinical trials? Basically, bottom line is what I really want to ask is, do the families report an issue with trust in terms of participating in these type of studies? If yes, uh, what could be or should be done differently to improve that trust so that more families would like to agree to participate in those clinical trials and research in like an acute NICU setting where you have to make a decision within hours, if not minutes? I don't know, Betsy, if you want, or yeah,
I can start it
Yeah.
out. That's fine. So to just touch quickly on the nurse inclusion, I think what's been really interesting over the past couple of years of working with a lot of you in this room and others as we, give input on study design. In neonatology, it seems like a lot of things are like building the plane as you fly it. So, I consult on a few different studies on behalf of our organization, and one in particular I'm thinking of where a lot of things that we talked about in planning, and then there's consenting issues like as we're going, and then we're changing those in real time. But really what could have helped is a more concerted effort of including nurses, of a more robust training, for everyone to be on the same page and really have people see the value of building health literacy in the moment. HIE is a great example. It's families that are coming into this are in a crash course into the NICU, right? The majority do not know that this is coming. And very often, families are separated. Mom's maternal health might be completely out of it, depending on if she had to have an emergency C-section and has her own health issues. I mean, we've seen families that have been enrolled in clinical trials that didn't know it was for HIE. 2 years out, which is just wild to me. But we're so thankful that those families also find it really important to give back, to give their perspective of what would be more helpful in those critical moments. I mean, for cooling, you have to be initiated within 6 hours. Anything that's an adjunctive with cooling right now is, again, very, very, very tight timing. It's not impossible. We've gotten this done. We've obviously have cooling because families opted into those tight time frames. So what do we need to do? Plain language, quick key messages. You can't, give someone 20 pages of a consent form and say, here you go. That's not going to help them. They need the quick messages. They need to know that you have to really come from a storytelling perspective of why we're at the point we are with whatever it is that you're trying. So with HIE in particular, we're looking at, we have a historical, why we have cooling is we have a historical opt-in from families that took the challenge to opt their children into this. It sounds like science fiction for those of you that have not, are not as familiar with therapeutic hypothermia. You're cooling your baby down for 3 days. It sounds crazy as a parent, but yet we had thousands of parents that have, opted in their babies to those trials. Why we have that as standard of care. But so that legacy, I think, is really important when you talk about mistrust. There's a lot of things that go wrong in our community, that some that can be prevented, some that cannot. So families are very mistrusting because they're in the midst of trauma of something not going right. Whether it's, again, baby stops moving at home or something goes wrong in labor and delivery. And they're like, wait, could that have been prevented? And do I trust you not? And we're in a huge era of misinformation and disinformation, as you all know. So, we have a lot working against us, but there's a lot of things we can do together to decrease these barriers and find success as we move these things forward.
Well said. Ashley, do you want to go?
So I am a family nurse practitioner. So when I was sitting in the NICU, it wasn't like a total shock. I didn't know enough to help, but I knew enough to know to read the monitor, like something's not right. But I would sit in the hours where an open bay NICU, and I would listen to and watch all the families come and go. And I realized that the families, not only were they numb, they were in shock. They had no clue of what to ask. The only question they would ask is, how is my baby doing today? And I'm thinking, like, that's not enough. You need to ask this, 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 this. But I'm trying to stay in my lane, in my little spot. And I also have a podcast where I interview different NICU moms and dads. and they just talk about their needs weren't being met as a family. So it's like, how can you ask more of me if I am numb, I'm shocked, I'm trying to figure out how I'm paying $25 a day for parking, I'm trying to figure out how much time to take off work. Do I take off work now? And which I went back to work 2 months into a 6-month NICU stay. Like, do I take off work now, or do I return to work now? So then when my baby comes home, I can be home with my child for a little while. So those are the more pressing things. How your four walls, how am I going to eat? How am I managing, like, and relationships fall apart in the NICU. And so you're trying to manage your core, and then you have staff that are asking more of you. So we have to figure out how to meet those needs, and then explain them in a way that they can easily digest. Because a lot of times you've already did rounding, you have a million people looking at you, you're trying to process that information, and then you have another person coming to you like, hey, do you want to sign up for this? So you have to make it more, like she said, digestible. Like, this is what we're going to do, this is what this means, and this is how we can apply to your baby and then also future babies. And then when you bring that in, I feel like more families will be more willing to participate.
Sounds good. Thank you so much, Ashley. And thanks, Betsy.
Kamaji, can I just add
Yes, go ahead.
I just wanted to say too, thank you both. I think this perspective was really key. These were a lot of discussions we had within the Seizure Work Group 2.0, because of course, from investigators and from drug development perspectives, everybody knows and can list all the challenges with acute consents and for acute symptomatic seizures and other time-sensitive trials that enroll in the NICU. And of course, then the next question always becomes like, well, maybe we should be thinking about exceptions from informed consent, because these are, acute families, are under such stress, and they're really not going to be able to consent in those situations. But I think this perspective is so important to hear, that it's not about that they're not capable. We have precedent for doing this for the cooling trials and other acute trials in the NICU. It's about really getting this input about how we can do that more effectively, rather than taking the choice away from families. We heard that very loud and clear during our work group discussions around that, should this be trials under EFIC, or these, should that be the answer to these challenges? And it was very clear that I think there's a lot we can do on the consent side and on engaging families and figuring out ways to make that digestible and still including them in the discussion around enrolling their child in a trial. So I just wanted to really emphasize that.
Thank you, Anne. Yeah, definitely relevant. So this is a question to, I guess, both Anne and to Janet, because it's relevant to both of you guys. There's a whole lot of significant amount of drug development work that happens in either in adults epilepsy or in like older children, pediatric, epileptic drug development, but not quite so much when it comes to neonatal seizures, for obvious reasons, for a lot of reasons. But I'm pretty sure there's a significant amount of learning that is to be had from I guess sponsors that routinely engage in older pediatric or adult epileptic drug development, right? What sort of but obviously it's not happening. I think it should happen. It could happen, but it is not. But what are those barriers in terms of what do you think, Janet, from like a I guess, NICU perspective, academic perspective, who also does a lot of clinical research, like barriers from extrapolating the learnings from older pediatric or adult epileptic clinical trials into neonatal setting, if there are. What are those barriers, and what will it take to break down those barriers, so that the type of, I know a huge, tremendous number of companies, pharmaceutical companies, get engaged in epilepsy research, but you would probably count the number of pharmaceutical companies on a single hand when it comes to neonatal seizures.
Yeah, so great questions. I mean, I think sort of that requirement to have, pediatric drug development for any new drug has been a help, but it still often leaves out the newborns.
Yeah.
I think the people who are interested in neonatal drug development are often basic scientists or physician scientists who recognize the need. And that's a pretty small group, and it's actually a smaller group because it's been very difficult to maintain a pipeline of pediatric physician scientists. That's been a major conversation within the society, like, for example, the Society for Pediatric Research, that tries to promote, support, develop pediatric physician scientist careers. So that's one part. But from the pharma side, I mean, I think it would be ideal if pharmacists, pharmacology companies are developing new drugs to at least do the animal work in neonatal models so that we can get some idea of the signal and safety in neonatal animal models and not just pediatric animal models. So I feel like, you know, when, and maybe the farm, we don't have anybody from farm up here, so maybe we should, but maybe some.
There's a
some
reason,
folks
right? Because the reason we do not have is because there are so few.
Right.
Yeah.
But anyway, maybe the pharma folks in the crowd can enlighten us a little further on that regard. So that's one challenge. So for example, you know, UCB came to me to say, how do we get lacosamide tested in newborns? And, you know, we end up doing this retrospective, very kind of weak study that's very preliminary, provides very preliminary data and doesn't really get us the answers we need. So that's from the basic drug development side. I think, you know, the physician scientists and the clinical researchers like myself very much want new drugs. We want better drugs. We don't just want to take a drug that is used in an older kid and try it in a newborn just because it worked in the older kid, because we know that that's not necessarily true. I do think we can learn something from our colleagues who work in acute seizures. So the groups that work in status epilepticus, pediatric and adult status epilepticus, because their trials are very similarly designed to our trials, i.e. they're acute seizures as opposed to epilepsy. So all of those epilepsy clinical trials that we run in children, I'm like always envious of my epilepsy colleagues. They just sit in clinic and the patient comes to them and then they're like, hey, we have this new drug. You want to give it a try? There's no like running to the NICU at 2 a.m. to try to talk to some family by telephone and fax them Sonia's nodding her head. It's like, it's ridiculous
So
what we do. So I mean, I think learning from the pediatric and adult trials, it really is sort of the trials of status epilepticus are the way to go. That said, I think a lot of them have gone to an EFIC model. They've gone to the exception from informed consent because these are emergencies and everyone recognizes the emergency because it's so much better defined in children and adults. So the reason we say status epilepticus needs to be treated within 5 minutes in an adult is because we have really good data that say, hey, if you don't get the drug in quickly, then it leads to a longer seizure. And we know that when you have X amount of minutes of seizure, it causes injury. We still don't have that defined for newborns. So we can't legitimately say, oh, we clearly need exception from informed consent for status epilepticus in newborns because we actually haven't really defined status epilepticus in newborns yet. So that's where our struggles are. And so I think, you know, engaging parent groups and having them weigh in on what's important to them is obviously key, but we're just not, I just don't think we're going to be able to go to the EFIC route until we can say we know exactly how much seizure activity is too much for the newborn brain because they don't have prolonged continuous seizures the way children and adults do.
Yeah.
So those are the two sort of main points. Obviously I could ramble on.
I could probably write a book on that, right?
I'm going to let Ann
Yeah.
take it.
You have a
further.
comment? Yeah, go ahead.
Again, I seem to be making a lot of comments to this. My apologies. Along the lines of neonatal models, clearly this is a challenge for folks in industry and academia also. But there's obviously been a push more recently by the FDA to look at non-animal models. And I don't know if any of our regulators can actually comment about whether there's ultimately going to be traction, especially in the neonatal space, using initially non-animal model testing to look at, you know, drug development at an early stage and at least look at mechanisms of action, etc. So anybody interested in commenting from?
We very much hope so. I'll say this is a rapidly evolving area. Obviously, there's been so much interest in a lot of recent workshops in the area. We were just actually having a conversation about this around, you know, there's guidances out there around developmental neurotox or developmental toxicity and, you know, the requirements around 2 animal models to assess those types of things. And that's, you know, in existence, and then we have a lot of new guidance or discussions around how we can incorporate these new approach methodologies or NAMs or other approaches to evaluate toxicity. And I was describing us almost in this slightly limbo area right now, where we're definitely interested in looking at those proposals and looking at that type of data to investigate some of these potential toxicity signals and things. We don't have a guidance out there to say this is the model or this is the approach that you should use, but obviously it's a very active area of interest in the scientific community as well as in the agency. So, you know, there have been a lot of recent workshops hosted by the agency, both internal and external, about how we should be thinking about this data and how we should evaluate it as it's coming in. But I'll just say that it's definitely an area of interest.
So just a follow-on comment. So then, from the drug development side, generally one gets, unless one has an, one's going after an orphan indication or has, is going down sort of a fast track, rare disease situation, you usually get one, possibly two opportunities for a pre-IND, but it's usually one. So there is some limitations on our end in terms of approaching the agency, because we can't keep on knocking on the agency's door and asking about this. How would you recommend us pursuing that? And again, it's evolving, so we'd have to work hand in hand with the agency and get more feedback and have an opportunity to have those discussions, not only in forums like this and at other meetings, but more formally, specifically for a specific target we're working on.
Sure. I'll make a comment, and then I'm going to phone a friend here because Lynn's in the back is smiling. But there are several programs, expedited programs, and programs designed specifically for rare diseases and orphan diseases that do allow for more interaction with the agency. So that's definitely a route that could go and have those opportunities to have more pre-IND, IND meetings or interactions with the agency. And those are probably the best bet to kind of increase your ability to interact with the agency in those kinds of discussions.
And neonatal seizures do count as an orphan disease, right? So
Yes,
in
there
that
are,
sense,
there
they
are
are
orphan
orphan
designations
disease
for
by
neonatal seizures as the condition.
by numbers, yeah.
Well,
Yeah, go ahead.
they sort that out. Two things. We were able, through collaborations, to deconvolute the question about animal model testing for tuberculosis drug development 10 years ago. And the first NAM for efficacy to be endorsed by regulatory agencies was completed 10 years ago, even before NAM was even a term. It was thanks to collaboration and the input and commitment from the regulators. So this is exactly the recipe for that. And what was the key foundational concept to make that happen? The fundamental question about what is the required predictive accuracy for the type of drug development question you need to answer. That was it. And we took all the data from the existing animal models, we rank ordered them, we found where the uncertainties lied that were unacceptable, and we looked at additional types of methods that could decrease that uncertainty, and that was the answer. If it could be done for an area that nobody cares about because it's a global health issue, not true, because we solved it back then. It can be solved for this specific question of seizures in the neonates specifically, which I think is a really important piece of the opportunity ahead of us. And if you're really interested in understanding more about the NAMS question and the evidentiary considerations that we're driving with the agency, come to the breakout session tomorrow at noon-ish.
Yeah.
on NAMS.
So, yeah, go ahead, John.
I'm really I don't know. There it is. Okay. Part of the challenge, I would think, for the drug developers, and Janet, you and I have talked about this before, is that it's such a heterogeneous population that results in seizures. And I don't know how you can expect one drug to take care of everything. Betsy was the one who sent me a number of papers on genetic disorders and HIE looking very similar. And we've started sequencing more and more of these kids with HIE, and I am just stunned at how many kids don't have HIE but have an underlying genetic disorder. And if I had my way, every baby with seizures would get sequenced. And then we're cooling babies that maybe aren't going to respond either. So I'm curious as to what you think is how do we select out the populations that these drugs are going to work on? And do we have the expectation that reducing seizure burden in a metabolic or an underlying brain disorder or something is going to have the same impact as it is from true HIE or not? And I'm also curious as to, what our parents tell us, the babies had no seizures for like 3 years or 6 years or 9 years. And I often hear people say, oh yeah, they had them as neonates and they're just coming back now. Well, that doesn't make a whole lot of sense to me either. I mean, maybe it does to you, but I'm
It
just
does.
curious.
Sadly, it does.
Yeah. So I'm just curious what your thoughts are.
I'm old enough that a baby I looked after as a newborn, when I was a resident who had a stroke, had his seizures come back in adolescence. And they were from that same stroke. They were not from something new. So it does happen. You can go that many years and not have a seizure. And then he actually had such refractory seizures, he went to epilepsy surgery. So doing great, but just to say, so yes, the answer is it can come back. So your point raises a couple of questions. So one thing that we also addressed in our Seizure 2.0 manuscript that has just been sent back with response to reviewers, is we did address questions of genetic testing, and that more and more we would advocate where reasonable to get whole genome sequencing. You may not be able to, have that information back quick enough to change management acutely, but certainly in the post-hoc analysis, it would help you with sorting out who actually had either a genetic epilepsy or a genetic contributor to their neonatal seizures. So that's one thing. Then with regard to the mixed groups, I mean, that was what I said at the very beginning, right? We do have these two large categories of newborn seizures, those with acute provoked seizures or symptomatic seizures, and those with genetic disorders, whether it's a genetic epilepsy or brain malformation, or the list of non-acute causes is incredibly large. There is reason to say, though, that some of the mechanisms may be similar. So although certain genetic epilepsies are, channelopathies, like, for example, we know that kids with KCNQ2 gene variants respond exquisitely to sodium channel blockers. But for the other things like brain malformations, it's not so crazy to think that some of those same drugs might work. And so it's not crazy to include them in those early phase trials. And I think we discussed that in our first seizure work group, that if we're doing early phase trials, we should definitely be including all comers, because you might just find that group of patients that respond really well to some drug like vigabatrin for tuberous sclerosis, infantile spasms and tuberous sclerosis, where, their response rate is just so much higher that we actually don't start with ACTH as we were, or steroids as we would for other kids. So I think that I think that there's still rational reasons to test all of the different etiologies, when we're testing a new drug, and also to test those drugs and all those different things. So both from the pharma side and from our side, to think globally about it, but then to get the right information with the genetic testing. I think one point that we haven't sort of touched upon, and I'm going to maybe digress so you can stop me if you don't want to go in that direction, is that one of the real problems that we're learning with neonatal seizure clinical trials is that they really can only be done at certain centers, right? This is not something where you can have the same kind of trial design at Boston Children's Hospital that you can in Uganda. We can't get we can get rapid whole genomes, for research reasons and even clinically within a week. But we're definitely not going to get that in many places in the US, let alone Uganda. And then the continuous video EEG monitoring, which really is needed for the gold standard measure of seizures. And so not only is that expensive to apply and to monitor, but there's not that many specialists that will be able to read those and interpret them accurately. I am hopeful that AI methodology is going to help us. We have seizure detection algorithms that are being improved. I'm working with a group right now that I hope will be able to help us improve this. And then that would really do away with the problem in Uganda. So then if you had a simple application of an EEG device, for example, a CAP or something like that, then you could bring your clinical trials to more places, maybe not rural Uganda, but maybe big hospitals in Uganda. And I just picked Uganda randomly, not for any particular reason, but just to say that you could imagine a place where this could actually be done and you could actually start improving the clinical care of patients globally and not just at the, quaternary children's hospitals in the US where all of the technology is available. Anyway, bit of a digression, but
No,
I still think
operational
germane to your
excellence,
point.
very important, definitely, infrastructure issues, crucially important. So
Actually, they are reporting now, even at Boston Children's, that you could get a very rapid turnaround in 3 hours, 3 to 4 hours.
Yeah.
So that's the next step.
So one last comment, Sonia, do you want to go next?
Sure. Sonia Wong from University of Minnesota. And I just wanted to share my experience from my clinical trial. We are funded by the FDA to run the NeoLev 3 trial, which is a dose escalation of levetiracetam and a control group of phenobarb. But my story actually touches upon all the topics that were just discussed. Just a couple days ago, a baby was born, severe HIE, and but this was upstate Minnesota. And then and the baby was transported early, and I got a heads up from the neonatologist very early on. And I spent the entire day trying to call the parents and couldn't get through. My research team kept running to the unit. And finally, by 5 o'clock, 6 o'clock or something, I get a report from the nurses that they also med flighted one of the parents. And then so I'm driving in at 7 o'clock, and my consent form is 15 pages. And I did the bullet points, but there are 4 places that the parent has to sign. One is, will they consent to future contact? One is for the actual study. Then there's a HIPAA form, and then there's can I get your email and your text message? And by the 4th one, the mom was rolling her eyes. Fortunately consented. And since we know sort of the trajectory of HIE, I knew that within about 4 to 5 hours, the baby was going to start seizing. It was my guess. And so I'm sitting here looking at my clock thinking, when can I sleep and when can I get up and watch the EEG? And so by 2 in the morning, yes, well, by 2 in the morning, sure enough, right around 24 hours, the baby started seizing, but it was so subtle. And I was so nervous because as a study patient, I actually woke up a colleague and said, hey, can you confirm this? And then I started tracking the hours. So the 12 minute per hour, seizure burden, our cutoff is 8 minutes per hour for this phase of the study. And so I'm sitting there calculating and saying, okay, at what point in time does this baby now need to come out of the study protocol? And then at 2:30 in the morning, I'm calling the pharmacy and arguing because they're not giving the drug because it's an investigational drug, not a standard drug. And finally, I just said, forget it. And then I actually just ordered the standard drug, which is the same thing. But I have to say, within 15 minutes of me ordering the standard drug was in the unit and infusing, and 5 minutes into infusion, the seizure stopped. And so it was beautiful to watch the levetiracetam take effect. And so I'm sitting there going, okay, how long do I have to stay up to watch this? And at 5, I say, okay, I just need 30 minutes of sleep or so. So I go to sleep for 30 minutes, wake up, and then and I start watching, and sure enough, the seizures start coming back at 6 a.m., but it's not 30 seconds, which is our criteria for treatment. And we're using a lot of the INC data from the past. And so as soon as we hit 45 seconds, I'm calling the unit, I randomize, and the baby goes to phenobarbital. So I treat with phenobarbital, and then within 5 to 10 minutes of phenobarbital, the EEG is just completely snowed, which is not what it did with the levetiracetam, and it stays snowed. And when I say snowed, I mean, I'm talking like burst suppression for only a standard dose of phenobarb, and it lasts for at least 6 to 8 hours. And so I'm thinking, is this overkill? And this is exactly why we need to do these studies. But the only reason this one baby could have been enrolled is and consented and intervened on is because I had a neonatologist who gave me a heads up 12 hours before. We had research staff telling me I was able to go in at 7 o'clock at night, and then I was able to stay up all night. And so we are working with a company to provide data for computerized algorithms, but this story just kind of emphasizes all the challenges that we've had. And then my last comment is I have 2 sites in the US, and consent has been very low for the 2 sites in the US. I have 2 sites in a foreign site, and their consent rate has been over 95%. So there is truly a difference in culture.
But just say what the site is, right? It's New Zealand.
Yes.
Right.
Yeah.
So a completely different public health culture and
Yes.
medical insurance culture and all that.
But our first NeoLove 2 trial had over 95. This was 2011 to 2017. That was over 95% also positive consent rate. So I just want to share that story because it's hot off the press, but it really does touch on all the topics that were discussed.
Yeah, I think I wanted to get into issues with respect to phenobarbital and how that impacts clinical research efforts, but out of time. The good thing is we're going to have a networking lunch break right outside, and we will resume here. And I think 1:15, exactly in 1 hour, to go over HIE related topics. So thank you. This was a really nice discussion.